Hello everybody, myself Divya, Botany faculty. Today our topic for discussion is sexual reproduction in flowering plants. When we say flowering plants, what do we mean by flowering plants? So when I say flowering plants here, we are going to be discussing about angiosperms. So flowering plants usually represent angiosperms and we are going to see how sexual reproduction takes place in angiosperms. So who is the reproductive part of the angiosperm? So flower is the reproductive organ or reproductive part. Now from where do I get flower? My shoot apical meristem to bear a flower gets converted into a floral meristem. So shoot apical meristem gets converted into floral meristem. So instead of bearing leaves, they will be start bearing flowers. So when we take flower, what does a flower have? So the stalk of the flower is called as pedicel. So the stalk of the flower is called pedicel. Now, if we take a cross section of the flower and see, so you have a condensed axis with a swollen base like this. Now this part here is the pedicel and the swollen portion here is called the thalamus. On the thalamus are the different whorls of the flower present. So there are four different whorls present in a flower, namely calyx, corolla, androecium and gynoecium. So the outermost whorl is the calyx, which is green in color. So this is the first outermost whorl, which is calyx. Now the unit of calyx will be sepals. The second whorl will be the brightly colored corolla. So this is going to be a second whorl. So this is my second whorl, which is corolla. Now the unit for corolla will be petals. The third whorl will be androecium. So this is our third whorl, which is androecium. The unit for androecium is stamen. And the fourth and the innermost whorl is your gynoecium. Fourth and the innermost whorl is the gynoecium, which has three parts, that is stigma, style and ovary. So this is the innermost whorl and the final whorl, which is gynoecium. Now the unit for gynoecium will be carpel. So out of these four whorls present here, calyx, corolla, androecium and gynoecium, the calyx and corolla are called non-essential whorls, whereas the whorls androecium and gynoecium are called essential whorls. Why these are called as essential whorls and the other is called non-essential is the calyx and corolla do not take part in reproduction, whereas androecium and gynoecium take part in reproduction, and that is the reason why androecium and gynoecium are called essential whorls. Now moving further, we will be discussing each of these essential whorls in detail. So the first essential whorl what we are going to discuss is the androecium. So what is androecium? Androecium is the male reproductive whorl. So this androecium unit is stamen. The androecium unit is stamen. So if I have to draw a stamen, the stamen will be like this. So the stamen has two parts present in them, namely the upper portion, which is the anther, and the lower portion, which is the filament. Now, where are these present? The stamens are present. They are present on the thalamus. So, this will be my thalamus. Now, when we talk about the filament, see the filament is attached to the thalamus at one end and to the anther on the other end. So, the end where it is attached to the thalamus, this end, where it is attached to the thalamus, you call that end as proximal end. And the end which is attached to the anther, this end we call it as distal end. Proximal means closeness. So where it is attached, that will be the proximal end. So the attachment of the filament is with the thalamus. So you call that end as proximal end. The end which is attached to the anther, you call that as the distal end. Now, are these stamens attached only with the thalamus? No. Some cases, okay, the stamen are attached with the petals. Attached with petals. So such condition, when the stamen are attached with petals, such condition we call that as epipetalis. When our stamen or the androecium is attached with the petals, you call that as epipetalis condition. Now, example for epipetalis condition will be Solanaceae family. Solanaceae family. Now, what about this anther? Now, if we look into detail of this anther, we say anther is bilobed. Each lobe dithecous, and they are tetrasporangiate. Now let's see in detail what are these terms. So if I have to draw an anther here with the filament, so this part here is one lobe. Let us assume this is lobe 1 and this will be the second lobe, which is lobe 2. So when I take a section of this anther and see, I get a typical butterfly-like structure like this. I get a structure like this. So now these are going to be my lobes. So let us assume this is my lobe 2 and this side will be my lobe 1. Now this point where you can see the junction, like now this is called the line of dehiscence, where anther is going to burst and release the pollen grain. So now there is an outer covering which is protecting this lobe like this. So here I have, then I have something here. Again I have from here to here, then from here to here. Now this layer here, you call these layers as theca. So for each lobe, other than the region of dehiscence, the thecas are covered. That is why each lobe there are two thecas. So you can see lobe 2, the left side is lobe 2 where two thecas are present. Same way you can see the right side lobe 1 where two thecas are present. So that is why we call them bilobe. Each lobe is dithecous. Now each of this lobe at the corners will have a bag like structure. Now this is what we call it as microsporangium. So this we call it as microsporangium in the corners of each of the ends. So how many microsporangium are there? 1, 2, 3 and 4. So that is why we call that as tetrasporangiate. So 4 microsporangium are there. Now this microsporangium will mature to form the pollen sacs. Now moving further, if we see the structure of microsporangium or the anther wall, layers of anther wall. So from the previous slide, we have seen the corner. So we are going to just take one half of the one side corner. So as in every other case, the outermost layer will be epidermis. The same happens here also. So let us assume this is the first outermost layer, which is epidermis. Now, if I have to describe the epidermis, so this is single layer here. So this is epidermis. These epidermises are thin walled structures. They are outermost, protective. They are single layered and parenchymatous. So for epidermis description, you can use a thing called POPs, which is protective, outermost, parenchymatous, single layer, POPs. So you can remember epidermis as POPs, protective, outermost, parenchymatous, and they are single layer. So moving further, the second layer is called endothecium. The second layer here is called as endothecium. So now this is the second layer, which is endothecium. Now the inner walls of this endothecium has some thickenings. These are cellulose fibrous thickenings. So this endothecium has alpha cellulose thickenings. Now this endothecium is mainly associated with dehiscence of anther, the bursting and releasing of pollen grain. Then the third layer, it is multiple layers. Now they are not single layer, there are 2-3 layers. These are called as middle layers. This is the third one, which is middle layers. So they are multiple layers and they are short-lived. And these middle layers provide nourishment to the developing structures. It is going to provide nourishment to whom? Whatever microspores which is going to be formed, which we'll be discussing at later stages. And the last layer or the innermost layer, which is most important, is the tapetal layer, is the tapetum. Now each of these tapetum cells are basically binucleated. These tapetum, the fourth layer, which is the tapetum, which is innermost, they are nutritive, binucleate. So when is it going to be, how it is going to be binucleate means the karyokinesis is not immediately followed by cytokinesis. That is when you have binucleate condition. So and this tapetum is also responsible for production of calase, compatibility proteins, then they are responsible for production of pollen kit, and as well as the most important, sporopollenin. Now we'll discuss how these are utilized in the further sessions. So I hope you understood whatever was being discussed today. Thank you.